Hello, everyone. It's a privilege to address you today on a topic that fundamentally reshapes our understanding of microbial evolution, horizontal gene transfer, or HGT. We often think of evolution as a gradual, vertical process, changes passed from parent to offspring. But in the microbial world, genetic information doesn't just flow downwards, it also flows sideways, across species boundaries. This lateral exchange of genetic material is arguably one of the most powerful and swift architects of microbial diversification and adaptation, and today we'll delve into its intricate mechanisms, the selective pressures that drive it, and its profound evolutionary consequences. To truly appreciate horizontal gene transfer, we must first contrast it with the more familiar concept of vertical gene transfer. In the realm of eukaryotes, and indeed for much of life, evolution is primarily understood as the inheritance of genetic material from a parent organism to its offspring, accumulating mutations over generations. This is the tree of life metaphor. However, in the microbial world, specifically bacteria and archaea, the tree of life is more akin to a web of life. Here, genetic information isn't confined to vertical lineage. Instead, it can be exchanged laterally between unrelated or distantly related organisms, often in a single generation. This is the essence of horizontal gene transfer. It's a process where genetic information bypasses typical reproductive barriers. The impact of HGT is truly immense. It plays a massive role in microbial evolution, allowing for astonishing adaptability to novel environments and facilitating the rapid spread of traits that confer immediate selective advantages. Imagine an organism, through a single event, acquiring a completely new metabolic pathway or an entire defense system. As researchers in this field, we often say that if vertical inheritance is the slow, steady accumulation of genetic change, the meticulous editing of a manuscript over centuries, then HGT is the rapid, explosive acquisition of entirely new capabilities. It's almost like a biological copy-paste function, allowing bacteria to instantly integrate complex, prepackaged genetic modules that can completely alter their phenotype. This makes them incredibly dynamic and challenging to track. Our first mechanism of horizontal gene transfer, HGT, is transformation, the direct uptake of naked, extracellular DNA by a bacterial cell. This DNA may come from lice cells or environmental sources. However, transformation isn't passive. The recipient cell must be in a physiological state called competence. Natural competence is genetically encoded and found in many bacteria, including Haemophilus influenza, Neisseria gonorrhoeae, and Streptococcus pneumoniae. These bacteria form specialized machinery for DNA uptake. Artificial competence is lab-induced, often in E. coli, using chemicals or electroporation. At the molecular level, transformation involves several key steps. DNA-binding proteins on the cell surface capture extracellular DNA. Translocation complexes, related to type II secretion in gram-negatives or pilus-like structures in gram-positives, pull DNA through membranes. In gram-positives, one strand is degraded during entry, the other enters the cytoplasm. Once inside, single-stranded DNA may be integrated into the chromosome via homologous recombination, mediated by RECA and other proteins. Competence regulation is highly controlled. In many bacteria, it's density-dependent, often regulated by quorum sensing. For instance, S. pneumoniae uses a competence-stimulating peptide, CSP, to trigger competence at high densities. In contrast, Bacillus subtilis activates competence under starvation, likely to gain survival traits. Ecologically, transformation allows bacteria to acquire new genes from dead cells, promoting rapid adaptation and the spread of traits like antibiotic resistance from the environmental gene pool. Conjugation is perhaps the most impactful form of horizontal gene transfer, HGT. Often termed bacterial sex, it involves direct cell-to-cell contact and one-way DNA transfer from a donor to a recipient. This process is mediated by conjugative plasmids, extrachromosomal DNA elements. The classic model is the F. fertility plasmid in E. coli. A donor cell, F, carries the plasmid, which contains TRA genes encoding the machinery for conjugation. Formation of a sex pilus, which attaches to the recipient. Pilus retraction pulls the cells together to form a stable, mating pair. A relaxase enzyme nicks one strand of the F plasmid at the origin of transfer, ORIT. The single-stranded DNA is transferred into the recipient through a type IV secretion system, an energy-dependent protein complex. The recipient replicates the strand to make it double-stranded, while the donor uses rolling circle replication to restore its own plasmid. A notable variant is the HFR, high-frequency recombination, strain, where the F plasmid integrates into the chromosome. During conjugation, chromosomal genes can also be transferred, allowing broader genetic exchange. Conjugation's significance lies in its ability to rapidly spread genes, especially those conferring antibiotic resistance and virulence, not just within species, but across species and genera. It bypasses phylogenetic boundaries, enabling fast adaptation. In research, resistance genes have been observed transferring between pathogens, drastically increasing their clinical threat. Conjugation acts like a bacterial file-sharing network, enabling instant acquisition of complex traits. The third major mechanism of horizontal gene transfer, HGT, is transduction, where bacterial DNA is transferred via bacteriophages. Unlike our previous focus on phage life cycles, here we highlight their role in bacterial gene exchange. There are two main types. 1. Generalized transduction. Occurs during the lytic cycle. When a phage infects a cell, it replicates and degrades the host DNA. Occasionally, fragments of host DNA instead of phage DNA are mistakenly packaged into phage capsids. These transducing phages inject the bacterial DNA into new recipients, where it can integrate via homologous recombination. Since the process is random, any bacterial gene can be transferred. 2. Specialized transduction. Happens with lysogenic phages that integrate into the host genome as a prophage. When exiting the chromosome to enter the lytic cycle, faulty excision may occur, and adjacent host genes are mistakenly included in the phage DNA. This hybrid DNA is then packaged and transferred during subsequent infections. It's specialized because only genes near the prophage site are transduced. Transduction's impact is substantial, especially for gene transfer among closely related species or strains, given phages' narrow host range. It's a key driver in spreading virulence factors and antibiotic resistance. A well-known example is lysogenic conversion, where prophages introduce new traits. Vibrio cholerae gains the cholera toxin gene from a prophage. E. coli O157. H7 gains the Shiga toxin gene similarly. Ironically, while phages are bacterial predators, they can also act as genetic couriers, inadvertently shuttling useful DNA between cells. While we've covered the mechanisms of horizontal gene transfer, HGT, how DNA moves, it's equally important to understand the vehicles that carry this DNA. These are called mobile genetic elements, MGEs, DNA segments capable of moving within or between genomes, driving bacterial evolution. Beyond conjugative plasmids, key MGEs include: first are the transposons, known as jumping genes. These sequences move between plasmids and chromosomes using the enzyme transposase. They're critical because they often carry accessory genes, including those for antibiotic resistance. Some of the earliest resistance genes were found on transposons. Their mobility allows them to jump onto conjugative plasmids for widespread or disrupt genes tied to virulence or function. Second, the integrons, genetic platforms designed to capture and express gene cassettes. An integron typically includes an integrase gene for cassette integration, an ATTI site for insertion, and a promoter for gene expression. Integrons are especially notorious for accumulating multiple resistance genes, forming multi-drug-resistant strains. Think of them as modular systems letting bacteria plug in new capabilities. Clinically, they pose a major resistance threat. The third are the genomic islands, large DNA regions, often hundreds of kb, acquired through HGT and integrated into chromosomes, often at tRNA genes. They show altered G plus C content and encode integrases. Genomic islands commonly carry clusters of genes for virulence, e.g., pathogenicity islands, resistance, or new metabolic functions, enabling bacteria to gain complex traits in a single event. MGEs are the architects of HGT, constantly reshaping bacterial genomes, adding new functions, discarding old ones, and rapidly accelerating microbial adaptation. Understanding MGEs is vital for predicting and controlling the spread of resistance and virulence traits. Given these sophisticated mechanisms, a natural question arises. Why does HGT happen? What drives it? HGT isn't random. It's often a direct response to specific selective pressures in the environment. In clinical settings, the presence of antibiotics is a major driver. Antibiotic exposure creates intense selection for bacteria that possess resistance genes. HGT allows bacteria to rapidly acquire such genes, often from nearby resistant cells, rather than relying on rare, spontaneous mutations. A bacterium can, for example, gain an entire resistance operon through transformation or conjugation in a single step. Host immune pressure is another strong selective force. Bacteria that acquire genes for immune evasion, altered surface antigens, or toxin production through HGT gain a clear survival edge within the host. Beyond host-related pressures, environmental stresses like nutrient scarcity, temperature shifts, pH changes, or heavy metal exposure also promote HGT. Acquiring genes for stress response or new metabolic pathways can be critical for survival in fluctuating conditions. Several biological factors influence HGT likelihood. DNA availability in the environment for transformation. Cell density, important for conjugation and competence induction. The physiological state of the cell, e.g., stress, can induce competence. Ultimately, HGT offers a powerful evolutionary advantage. It enables bacteria to quickly acquire adaptive traits that might otherwise take millennia to evolve via vertical inheritance. HGT makes bacterial evolution agile, allowing microbes to thrive under new or hostile conditions and contributing to their resilience and success across environments. The consequences of horizontal gene transfer, HGT, are profound, driving genomic plasticity and rapid adaptation in bacteria. Unlike eukaryotes with relatively stable genomes, bacterial genomes are dynamic, constantly gaining or losing genes. This ongoing genetic flux means bacterial genomes aren't fixed blueprints, but adaptive platforms, rapidly reconfigured in response to environmental pressures. This enables bacteria to exploit new niches and survive in extreme conditions with exceptional speed. Let's explore the key evolutionary impacts. First, the rapid adaptation. HGT allows bacteria to acquire entire metabolic pathways, enabling degradation of novel pollutants or utilization of new carbon sources. They can also gain genes for survival under extreme temperature, pH, or salinity, expanding their ecological range. Next is the antibiotic resistance. Clinically, this is the most alarming outcome. HGT is the primary driver of multi-drug resistance spread. Rather than evolving through single mutations, bacteria can acquire full gene cassettes, transforming treatable infections into life-threatening ones almost instantly. Then we have virulence evolution. HGT fuels the evolution of pathogenic traits, such as through pathogenicity islands, large DNA segments encoding virulence factors acquired in single HGT events. And last is the redefining species and the pan-genome. HGT challenges our concept of bacterial species. Rather than a fixed genome, bacteria have a pan-genome, the total gene set across all strains of a species, including both the core genome, shared by all, and accessory genes, acquired via HGT. This makes bacterial evolution resemble a web or network more than a tree. In essence, HGT is central to microbial evolution, blurring species boundaries, enabling instant innovation, and complicating our efforts to control pathogenic bacteria. Understanding its consequences is critical in both ecological and clinical contexts. Let's zoom in on one of the most critical clinical consequences of HGT, its central role in the rapid spread of antibiotic resistance, AMR. This is not theoretical, it's a driving force behind the global health crisis we face today. HGT is the primary mechanism for the dissemination of resistance genes across bacterial populations, both within and between species. It spreads resistance far faster than spontaneous mutation. Consider some notorious resistance genes. Beta-lactamase genes, BLA genes. These enzymes degrade beta-lactam antibiotics like penicillins and cephalosporins. Yes, BLs, extended-spectrum beta-lactamases, and carbapenemases, e.g., KPC and DM1, are often found on mobile genetic elements, enhancing their spread. MCR1 gene confers resistance to colistin, a last-resort antibiotic. Its discovery on a plasmid in China, 2015, raised global alarm. It meant colistin resistance could now spread via conjugation. NDM1 New Delhi metallobeta-lactamase 1 found on highly mobile plasmids. It provides resistance to nearly all beta-lactams, making it one of the most dangerous AMR genes known. The speed and scope of resistance gene dissemination stem from conjugative plasmids and transposons. A single plasmid can carry multiple resistance genes and move between many bacterial species. If transposons are also present, resistance genes can jump to other plasmids or chromosomes, locking them into the genome and magnifying spread. This creates a cascade effect, accelerating the emergence of pan-drug-resistant bacteria, strains resistant to nearly all known antibiotics. This leaves clinicians with no effective treatment options, making formerly routine infections potentially fatal. The persistence and mobility of these resistance elements make AMR incredibly difficult to contain, highlighting why a deep understanding of HGT is crucial for developing effective control strategies. Beyond antibiotic resistance, HGT is a major driver of virulence evolution in bacterial pathogens. It allows bacteria to rapidly acquire entire toolkits for pathogenicity, transforming benign commensals into virulent disease agents. A key example is the acquisition of pathogenicity islands, PIs, large DNA segments, often tens to hundreds of kilobases, acquired via HGT and integrated into the chromosome. These regions serve as hotspots for virulence genes and often show hallmarks of foreign origin, atypical G+C content, flanking direct repeats, and mobility genes. PIs commonly encode adhesins, help bacteria attach to host cells. Toxins, damage tissues, or disrupt host functions. Secretion systems, e.g., type 3 or 4, inject bacterial proteins into host cells. Iron acquisition systems, to scavenge vital nutrients from the host. Classic examples include Shiga toxin genes in E. coli O157, H7, acquired via phage-mediated transduction, converting a commensal strain into a deadly enterohemorrhagic pathogen. Type 3 secretion systems in Salmonella, located on PIs, enabling host cell invasion and intracellular survival. Cholera toxin genes in Vibrio cholerae, carried by the CTX lysogenic phage, which transformed a harmless ancestor into the agent of cholera. These examples highlight how HGT enables bacteria to acquire complex virulence traits in a single step, bypassing the need for gradual evolutionary changes. By borrowing pre-assembled virulence systems from other microbes, bacteria can quickly adopt new infection strategies, enhancing their fitness and expanding their pathogenic potential. In short, HGT doesn't just fine-tune bacterial function, it can fundamentally redefine what a bacterium is capable of, making it a key force in the emergence of new infectious threats. To truly grasp the evolutionary consequences of HGT, we need to introduce a relatively newer concept in bacterial genomics, the pan-genome. For a long time, we thought of a bacterial species as having a relatively fixed set of genes. However, with the advent of high-throughput sequencing, we quickly realized that this isn't true at all. The pan-genome of a bacterial species is defined as the total set of all genes found in all strains belonging to that species. It's typically divided into two main components. The core genome. These are the genes shared by all strains within a species. These typically encode essential housekeeping functions and define the species' fundamental biology. The accessory genome, or flexible genome, these are the genes present in only some strains, or even unique to a single strain. This accessory genome is where HGT plays its dominant role. It includes genes for virulence, antibiotic resistance, metabolic adaptations to specific niches, and stress responses. HGT is the primary driver of the size and diversity of this accessory genome. Every time a bacterium acquires a new plasmid, transposon, or genomic island, it adds new genes to its accessory genome and, by extension, to the species' pan-genome. This means that two strains of the same species can have vastly different genetic complements, leading to significant phenotypic differences. The implications of the pan-genome concept are profound. It helps us understand the true diversity within bacterial species, why some strains are virulent while others are not, and why some are resistant while others remain susceptible. It fundamentally reconceptualizes what it means to be a bacterial species, moving away from a rigid definition to one that embraces the constant flux of genetic information. This concept is vital for tracking pathogen evolution and designing strain-specific interventions. To conclude our discussion on horizontal gene transfer, let's recap the key takeaways. HGT is not just a biological curiosity. It is a powerful, omnipresent force in microbial evolution. Through mechanisms like transformation, conjugation, and transduction, facilitated by a diverse array of mobile genetic elements, bacteria can rapidly acquire and disseminate genetic information. This enables their astonishing adaptation to new environments, drives the swift spread of traits like antibiotic resistance and virulence, and fundamentally shapes the very definition of a bacterial species through the pan-genome concept. Research in HGT remains highly active. We are constantly developing new genomic and bioinformatics tools to better track HGT events in real time within clinical and environmental settings. We're also exploring its intricate role in environmental microbiology, where it contributes to nutrient cycling and ecosystem stability. Looking to the future, the challenges and opportunities related to HGT are immense. From a public health perspective, understanding and ultimately combating the spread of antibiotic resistance via HGT is one of our most pressing global challenges. From a biotechnological perspective, we are learning how to engineer HGT, or its components, for beneficial purposes, for instance, in synthetic biology to introduce new functions into microorganisms for bioremediation or industrial applications. This ongoing genetic exchange is a truly fundamental aspect of microbial life. As a microbiologist, I can confidently state that it is both a formidable challenge to infectious disease control and, paradoxically, an incredible source of biodiversity and a powerful tool for those of us working in synthetic biology and beyond. Thank you for your attention. I am now happy to take any questions you may have. If you like this video, don't forget to like, share, and subscribe.